So for instance, if someone is like sick, for instance, using this technology, maybe if you do like an x-ray or some sort of like uh, body imaging, the cells that are particularly sick could then maybe translate a particular signal, whether it's a light, since you mentioned fluorescence, and then you can tell people, okay, maybe that's an illness, this is a particular illness in that particular cell. And by doing that, it actually helps them like map the location where the sickness is, particularly maybe in cancer, for instance. They can, using this technology, they can do a full proper mapping of where the tumorous cells are. Because sometimes I think the issue that happens with cancer patients is that sometimes even if they go for an operation, sometimes the doctors or the surgeons might miss some cells that are actually quite tumorous. So they do take out the huge chunk of the tumor, and then they miss a few cells that are tumorous, and then it kind of brings back the cancer again. So using this technology, we can then help, like in terms of like increase the efficacy of like surgeries, things like that, or like or even to just monitor how the disease is regressing as well. So this is actually quite exciting. And I don't know, I hope I've summarized it in the correct way this time.
Yeah, I think. Sorry, I think a bit of the focus is mostly like on the monitoring thing of like seeing the progression of disease, basically of like how cells change. Yeah, I think like that's also really important. I think Nika, you also.
Yeah, what you explained or summarized was actually really accurate in terms of possible applications. But there's another another exciting part in a different way, which is in molecular recording. You can track sequential events. So we can say, okay, there was an increase in this protein, which happened for some time, and then it happened again, and then it happened again. And this could be a different signal than just the presence of this protein or this increase. So it can potentially provide a more reliable signal, or combine different signals to say, okay, this protein was present, and the second one and the third one, which probably means that the disease is also present in that specific cell.
Welcome to the MedTech 101 podcast, and today we're discussing something that's rather micro than what we normally talk about. Usually we tend to focus on big devices or maybe implants, etc. But today we're going down to the genetic level of what makes us human, and how certain things that have been discovered recently, which allow people to use DNA, RNA, is like storage units, like basically having like a couple of cells as your USB device, to simplify it. And yeah, so today I'm joined with two master students out of Munich, especially the Technical University of Munich, which is quite a highly esteemed university. I mean, before after I finished my degree in England, I did consider applying to TUM, but I never told you guys. But I didn't apply in the end,
Yeah.
because I decided to go into industry, and I got a job offer from Johnson and Johnson. So I was like, should I work for Johnson and Johnson, or should I go to university again? So I was like, no, let me just go work. So today I'm joined with like Felipe Navarro, who basically is studying like responsibility in science, engineering, and technology. Like that's the full word of the
Yeah,
master's
that's a
degree.
full long name.
Yes. And I'm also joined with Nika Kobetic, who's studying like biochemistry, like, and these are all master students. So thank you guys for coming on the podcast today.
Yeah, thank you for the invite. Yeah, I don't know,
Nice.
like we're really excited.
Perfect. So, like, just to kickstart, then, like with your backgrounds and stuff, like, what is it like now, like studying in university? And I understand that you guys are kind of teamed up to sort of like work towards a project. So you've got people from different backgrounds and stuff, and I'm kind of glad that I was introduced to you, both of you, by my colleague, who, like one of my scouts colleagues I met about 9 years ago in Japan, and you guys are here today. So, what could you tell us briefly about your backgrounds, just for the guys listening. And we'll start with Nika. We can start with Nika first. So you go first, Nika.
Yeah, I come from Slovenia originally, where I did my studies up to my high school degree, where I also did the international baccalaureate diploma. So I was already familiar with the international part and using English in my studies. And then I did my bachelor's degree in biochemistry at the University of Tübingen. And now I'm doing my master's in biochemistry in Munich.
Nice, nice. No, that's good. And Felipe?
Well, I'm born and raised in Chile. I did my studies in molecular biotechnology. Back in Chile, I did my thesis. After I finished my degree, I worked for a year in this lab of free technologies with some friends and people that I esteem quite a lot. And now in October, I just started this master's here in Munich at TUM. It has this long name about like responsibility in science and engineering and technology. And basically, its idea is to uh, like actually consider all these emerging technologies and have a debate on like how they can impact society, how can like we actually engage with the technology so they're more democratic, they're safer, they're more inclusive. And I don't know, like for me it was something that felt really appealing when I applied, because like as someone that like was working in biotech all this time, it's like we are in the lab doing stuff, and we also often have these like conversations on like what could science look outside the lab, or like who's going to reach. And I don't know, like for me, this is like a way to go from those conversations towards like I don't know, like meaningful action in some sort of way. So
Nice,
it's nice,
nice.
yeah.
Nice, that's really good, and I'm just happy to see how like you guys have are international students as well. I mean, I was also an international student, and I think it's also quite nice when you go somewhere like away from home. Like it stretches you as a person. It helps you understand maybe new cultures, new ways of things. Like as we are discussing briefly before with the call started about how people sometimes maybe wake up at different times and different cultures, etc. And it's quite interesting, and it's always been really interesting because I think before in the past the technology hadn't really advanced to the point where like biological stuff, let's say on a genetic level, would start getting links to like direct sort of like technology. But I feel like the gap between biology and technology is kind of merging a lot more compared to what it was in the past. And yeah, and that's why we're here today. So, and you got an upcoming competition, the iGEM competition. Like what exactly is it for people listening? How do you go about saying it? And like any one of you can answer that.
Exactly. It's a competition in synthetic biology. So it's about repurposing the systems, the natural systems in biology for specific uses. And it can go into different applications: climate crisis, therapeutics, diagnostics. And it's about innovative ideas and innovative ways of using emerging technologies in synthetic biology. And yeah, the team is quite diverse, I would say. There's a lot of, we have biochemistry students, bioinformatics, molecular biotechnology. So we get a lot of different perspectives on the same problem.
Nice. And just to quickly ask there, like, how did you guys choose the teams? How did you? Because I know you guys are representing the university. So how did the team actually get formed?
Well, the IGEM Munich, there is like the Munich team that is going into this competition. Actually, like the people from previous year did the whole selection process. We're a bunch of students from TUM and from LMU, that's like other important German university here in Munich. And I think like basically we went to this like whole process of like discuss like understanding what iGEM is, what it implies, like wet lab competitions, dry like there's a part of wet lab, there's a part of dry lab, there's a part of human practices, there's like it's not just like you're doing a project in the lab, it's like it conveys a longer array of things that are involved. And I think like during that, and we had like this very like application process where like we put our interest and people got selected. And I think like I don't know, like I like the team we have right now. It has a nice dynamic, and
Nice.
it's yeah, people from different backgrounds, people that speak different languages. I mean, we use English, but like we speak different languages. We're like from different countries. So I don't know. It's really nice. It's really nice.
Yeah, I can imagine, and it sounds like the selection process is actually quite nice, and it's good. And where is it gonna be? Just to for people interested, where is the competition gonna be this year?
The final jamboree is going to be in Paris in October,
Nice,
where all the teams are going to present their work and yeah, the whole process of what went into it. And also, as Felipe mentioned, there's part in education, outreach, human practices, where we talk to both the general public and also experts in their fields to improve our project and also to promote and explain synthetic biology and yeah, get it closer to the people.
nice, nice. Having said that, now I'm kind of tempted to want to come. So it'll be nice. I'll be putting the dates and the links of all this like of this particular competition and the dates in Paris, etc. In case there's someone listening who wants to drop by and just see what you guys are doing. And if I'm free, is it happening over the weekend, or do you know when exactly it's happening? Is it during the week or over the weekend? Because if it's
Yeah,
happening over the weekend, that's been perfect. I'll try to come and see what you guys are doing.
I think the competition is okay. Is from the 23rd, something like that, of October. The Grand
Yeah.
Jamboree in Paris. I mean, like I would say it's the culmination of the competition in terms of like I mean, we're already doing stuff in the lab. We're already like working on the project, but the presentation of like the results like culminates in this like jamboree in Paris. So I think like it's also like something that is not only like, oh, we're gonna work a month near October and that's it. It's something that like I don't know, like we've been working since March, and it's a bit demanding in a good way, but it's not like something that you take easily. And I think like also that forces everyone in the team to bring up their best
Nice.
skills. I don't know.
Nice.
But yeah,
And
it's quite demanding. I think Virginia can tell you, like, yeah.
no, that's good. I'm sure it's gonna be exciting. But yeah, then like just getting the introductions out of the way. Now to actually sort of like get into the sort of like the points of this whole episode today, which is quite fascinating, is basically RNA based recording. And this is basically as I alluded to earlier, it's using RNA to like write code in the sense that to make it into Morse code, which can then be used to keep data from a binary point of view. Like what would you say about that? Like in a short, sort of like layman terms of what exactly you guys are trying to do.
Yeah, I can start. So essentially, the project idea is to use RNA for molecular recording, so to couple RNA editing to molecular events. This is a foundational advance, so it's a foundational project, and the uses of that are not in the forefront of our mind yet. It's more about using a new system, a new technology to do that. And we do that by editing RNA with something that is called um, an ADAR, so a domain that deaminates and changes the code briefly to then couple that to for example, a fluorescent signal. So then we can get a visual signal of a certain molecular event.
Nice.
Felipe,
And
you
just
can.
and just
Yeah.
yes, and just sorry, Felipe, just to try simplify that for people and correct me if I'm wrong, is that basically like RNA or DNA's got what they call like building blocks, right?
Yep.
And I think there's like four building blocks in DNA, or five. Is it four or five? I think it's four, right?
Four.
And
Four.
and what they do is each one they assign it a particular number, right? And if you know like how binary code or how binary numbering system works, it's ones and zeros. So by aligning them in certain ways, you can create this sort of like binary exchange. And so if someone looks at the RNA in the future and they see all these four building blocks, these are basically keys that they can then translate into numbers, basically. And this is done using what you call an ADA, which then translates that. Like, feel free to correct me if I'm wrong,
Mm,
but
Yeah.
Yeah, our project is more. Sorry, Felipe.
Yeah.
Just real quick, our project is more focused on molecular recording instead of molecular
Nice.
data storage. Molecular
Nice.
data storage is also a very exciting field because it's really
Yeah.
dense. You can pack a lot of information into very little volume. But actually,
Yeah.
RNA would be a worse option for that than DNA because it's less
Well,
stable.
makes sense.
And our idea in that was to use that more in the sense of not as a long-term data storage, but more
Okay.
to connect cellular events that are happening that are dynamic, and then connect that to a signal.
Oh wow. So this basically can then be used by humans or not? Is that
I,
the aim?
I think we are trying more to like provide the proof of concept. Like some other applications are actually probably going to be in the medtech field, and because the idea is like this idea of turning the cells into these like living like tiny recording devices. Like we can actually track molecular events, like things that are happening in the body, like basically like putting like a zoom camera and trying to understand what is happening like at the molecular level while it is happening and try to track it so we can actually like I don't know like if there are signs of disease or there are signs of modification or like changes happening in the body that are like relevant in biological terms, you can actually track it. And we decided to go with RNA. Basically, I would say because of two things. Well, basically, I mean, you have this idea where like DNA produces RNA, and this RNA then is translated into proteins. This like dogma of molecular biology, which are like a lot of the field is based upon. And I think we like we are moving towards RNA because like for me, it's two things. Is one, doing DNA modifications is a whole other thing. Like modifying the DNA to record these events implies like changing the DNA itself, whereas the RNA is a transient molecule that is like produced to be used and then is degraded. Like it has a really so this RNA is like this modification we're doing is actually transient and it actually allows you to like follow molecular events while they're happening, but it's not like a DNA modification. And also RNA is just like this messenger of information that is spread across the cell. So it's actually not in the nucleus, like, because the DNA is safely stored in the nucleus, whereas the RNA is present in the cytoplasm. So by tracking the RNA, we also have the advantage that we're like actually looking at what's happening in the whole cell and not just like what's happening hidden in the nucleus. So like that in that way, we think we can track molecular events in a better way. And yeah.
Nice. So actually, just to rephrase it again, what I said to simplify it for people, what you guys are basically doing is you found out that using RNA, you can basically track in terms of what's happening on a cellular level in someone's body. So, for instance, sorry, I dropped my pen. So, for instance,
No
if
way.
someone is like sick, for instance, using this technology, maybe if you do like an X-ray or some sort of like uh, body imaging, the cells that are particularly sick could then maybe translate a particular signal, whether it's a light, since you mentioned fluorescence, and then it can tell people, okay, maybe that's an illness. This is a particular illness in that particular cell, and by doing that, it actually helps them like map the location where the sickness is, particularly maybe in cancer, for instance. They can, using this technology, they can do a full proper mapping of where the tumorous cells are, because sometimes I think the issue that happens with cancer patients is that sometimes even if they go for an operation, sometimes the doctors or the surgeons might miss some cells that are actually quite tumorous. So they do take out the huge chunk of the tumor, then they miss a few cells that are tumorous, and then it kind of brings back the cancer again. So using this technology, we can then help, like in terms of like increase the efficacy of like surgeries, things like that, or like to, or even to just monitor how the disease is regressing as well. So this is actually quite exciting. And I don't know, I hope I've summarized it in the correct way this time.
Yeah, I think. Sorry, I think a bit of the focus is mostly like on the monitoring thing of like seeing the progression of disease, basically on like how cells change. Yeah, I think like that's also is important. I think Nika, you also.
Yeah, what you explained or summarized was actually really accurate in terms of possible applications. But there's another even another exciting part in a different way, which is in molecular recording. You can track sequential events. So we can say, okay, there was an increase in this protein, which happened for some time, and then it happened again, and then it happened again. And this could be a different signal than just the presence of this protein or this increase. So it can potentially provide a more reliable signal, or combine different signals to say, okay, this protein was present, and the second one and the third one, which probably means that the disease is also present in that specific cell.
No,
Yeah, and
this
just
is amazing. Yeah, continue,
yeah.
Felipe. Sorry.
Just to add a bit to that, like to do that, we're actually using this dead Cas9. This dead because it's not active Cas9. That is, it binds to this ADAR thing that Nika mentioned, and basically it has this idea of like the dCas9 allows us to like actually find the event, like, oh, like this happened, and we go to sequence and find it, and then this ADAR like actually does like leaves the mark in a way that is this deaminase that actually changes one adenosine for inosine, like this like molecular
The
one base
chain.
pair, very specific, very small change. So basically, it's
Nice.
like it's this complex that is like it finds and then it changes, and then yeah, this idea that we also have to do it like to count events and to have this like kind of like a traffic light in terms of like if it occurs one time you have a certain signal, and then if it happens another time you have another color like fluorescence, and you can actually like record these processes in real time. I kind of like the differentiation states of the cells, so
Yes.
like you can actually see like, oh, like it's actually advancing. So yeah.
And just to just take a step back quickly to
Yeah,
actually
sure.
just talk about what CRISPR is for people who don't know, for people who are listening,
Hmm.
like in a simpler way. CRISPR is basically I always like to make things into examples that like maybe an 8 year old will understand. It's basically like a copy paste function, like a copy cut paste function. Like in like so I would like you to imagine your DNA being this long chain, and the CRISPR can follow this chain and say, okay, here maybe we need to cut this, and it kind of cuts it, or maybe it changes the DNA there. So, and that's why people are kind of excited about this. And I think recent Nobel like prize have been won with the CRISPR technology, because it has the potential to like cure cancer. It's got the potential to change some the DNA of some viruses, like the HIV virus in people's bodies, etc., and make it actually sort of like useless essentially. So the capabilities are long, and then it was, but because of that, like any great thing comes with great responsibility. So people are worried that it also has the capacity to start changing people, creating these superhumans, etc., and things like that. Though no one has really been able to pull that off. I think there was a doctor in China who tried to cure like HIV in
Yeah.
tweens or something like that using this kind of technology. So the rabbit hole of this technology like goes deep, right? But that's basically what Philippe is talking about today. To say that using like the dead version of the CRISPR. I don't know if it's Cas18 or Cas9. Is it CRISPR Cas18 or Cas9? Which one is it particularly?
There's a couple of the Cas9 is the protein, the nuclease, which means the protein that cuts the DNA or the RNA. And there's a couple of different versions of the Cas proteins now. We're actually using the Cas13, not the Cas9, which is the most famous one. Yeah, they're a bit different into which molecules, which motifs they recognize, but yeah.
Nice.
But
Yeah.
basically, yeah, they're all part of this like big family of CRISPR-Cas systems that is like this system that like find sequences, very specific sequences based on an input, and then they cut it like this, like find and cut. And they can also be used to delete certain sequences that might lead to disease, or insert certain sequences that might be beneficial. But yeah, and also it's like this idea of like gene editing has been happening in like molecular biology since like, I don't know, the 70s, the 80s. But like with CRISPR, and that's why Jennifer Doudna and Emmanuel Charpentier won the Nobel Prize a few years ago. CRISPR allows a degree of specificity that was not seen before in the field, and I think like the easiness in terms of like handling of this in the lab, it's wild. It's way more easier than it was like, I don't know, like 10 years ago, 15 years ago. And yeah, I would like, even though there are like these examples from like this like CRISPR baby that happened in China, I think like there's also an acknowledgement that the technology is not there yet to be able to, oh, like we're gonna have like, I want to use CRISPR to have blue eyes, I want to CRISPR to be really strong, because like I mean there are a lot of things that like why I don't think that could be happening. One is like, I think the scientific community and people and policymakers, like I think like everyone is getting aware of like the ethical implications of the free use of this technology. And beyond that, I also think it's important to understand that the technology is not there yet. Like CRISPR, even though CRISPR is really really useful, as any other technology, is not perfect. It like they discovered that like in a very low amount, it like sometimes it does like off target modification. It's like it modifies things that you don't want it to modify. And like, so it's like still not deemed safe for like to do like this massive gene changes in humans. And also, I don't know, like it's like this idea of like there's a gene in my body that is like in charge of my hair color, in charge of like if I'm smart, like if I change these two genes I can get into Harvard or whatever. I think like that is like, yeah, like we're more than that. Like as humans, like the DNA is part of what makes us, but like it also is like the environment, and like how we're more than just genes, you know.
Of course. I agree with you. And just to add on to that, really, in terms of
Yeah.
this, they call them alleles. That's what they call them in genetics, where the ones that chooses which you're gonna have blue eyes or brown eyes, etc. But to simplify it for people listening, so basically, the human body has chromosomes. So we have like what they call like 23 pairs of chromosomes, like in a normal person. Some people might have more, some people might have less, but there's like 23 chromosomes that are paired. And the idea is that maybe on chromosome 17 in this particular small line, that's what's changed, that's what's giving me my eye color or something like that. So people then want to go at that basic level and say, let me restructure the alphabet a little bit to make myself smarter, taller, athletic, whatever it is people's desires are. Of which, if it did happen, it will create another problem in terms of fairness and like just our talent to be passed down to people, etc. But it's exciting, like you say that you're using this technology, and you guys particularly have chosen to use RNA, which is even which is smaller than DNA. So to like DNA is like is basically like a ladder, right, with two sides and it's connected, but it's like a helix, right? It's like a twisted ladder in simple terms. But the RNA is basically just one like side of the ladder, essentially. That's much smaller. And why have you guys chosen to why have you guys chosen RNA instead of DNA, for instance? Is it easier because it's smaller to encrypt this kind of messages in cells, or why specifically that?
So one was the reason that Felipe already mentioned about being physically closer to the molecular processes happening in a cell. So most of the metabolism, most of the reactions happen in the cytosol. So also most things that we might want to detect are in the cytosol. And we lose a bit of time if we write something on the DNA and then it has to get transcribed into RNA and then translated into proteins, the central molecular dogma, as we mentioned. So yeah, that is one of the primary reasons.
Nice.
And I
And
think
just
to
yeah,
actually,
sorry,
oh yeah,
sorry, Felipe, go on, it's okay.
I know, like I think like also is this idea of like like DNA exists, but like RNA, since it's the messenger, it actually tells your cells what they're gonna do in a sense. So it's like you can have the instructions to do it, but the actual like going and saying to like the proteins or to like different, like, I don't know, the mitochondria, or like, hey, you actually need to do this. Our RNA is in charge. So it's actually like it's a more, in scientific terms, it's a more like accurate description of what is really happening. It's not like, it's like actually like what book you're reading in your shelf, because like you might have like 10 different books, but which one are you actually reading? So it's like by choosing to focus on like what is actually uh, their actual instructions, you can actually see, you can focus on what is happening and know what has like the potential to be occurring in the cell.
Exactly.
Nice.
That connects again to the CRISPR blue eyes idea, because even leaving ethics completely aside, which we we shouldn't do. We should always look at the problem whole. But looking at the purely the genetics, it's much more complicated than most people think. There's also epigenetics, which is which genes actually get expressed. So it's not just what is in the genome. There's a lot of factors. So yeah, it's very important which parts of the DNA actually gets to the RNA level. And then there's also, okay, which RNAs get translated. So.
And just to quickly add on to that, for people to also understand that sometimes it's not a simple matter of just editing the genes, because even sometimes when you edit the genes, because let's say when we outside, let's say in the sun, because we have this UV rays, for an example, like this UV rays, sometimes they damage our DNA, but the body has like auto correction methods. I don't know how many number of ways the body corrects its own genetics, but I know it's quite a lot, and more methods that keep getting discovered one after another. And that's why sometimes in cancer, when we sometimes with cancer, because the body is now kind of lost that method of correcting its own genetics, and so in these so-called mutations that maybe lead to different eye colors, or like on a basic level, but sometimes these mutations are the reason why maybe like Michael Phelps was winning the Olympics so many times, because his body was producing like half the level of like lactic acid, just because he had this mutation from birth. Sometimes you can't really choose how the genes are activated. And you mentioned something that's quite interesting there, epigenetics. Like epigenetics basically is um like 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 for example, it's like what genes get expressed under what environment. So some people are saying that you moving to a different part of the world can activate certain genes in you that maybe which wouldn't have been activated were you somewhere else, and that can have a huge impact on health and things like that. And yeah, it's quite exciting. And I know you mentioned the ethical parts of it, which we're going to come to shortly. But I just wanted to quickly touch on like you mentioned the advantages of using like RNA in this particular case and stuff. Like looking at the technology itself, is it easily accessible, or do you need like special lab equipment for you guys to do this kind of work, just to give an idea of of how easily can this be replicated around the world, etc. Do you need special equipment, or is it something that's easy to do?
I mean, I think I would say our current project is, since like iGEM has these different, like as Nika mentioned, these different villages, like different areas that you want to do. Like in the process of development of our project, we became really interested in this idea to track molecular events, and we ended up going with foundational advance, that is, um, and a village that is a bit different from what iGEM, like all the other iGEM um, teams that are mostly focused into like explicit, very concrete applications. Ours, I think, is more like trying to provide this like molecular tracking tool that can be actually, like, for example, be then like used in medical fields. I think like what we really like, for example, is that the way of visualizing it and choosing to use fluorescence to visualize it, it simplifies the process in terms of like you can actually see it with, like, I mean, you need a fluorescent microscope that is like this UV microscope where like you can actually see the fluorescence, but it's more like it's like this like UV light like in the clubs or whatever, but but in a cell level, uh, but like, I don't know, like the other methods that currently exist require sequencing, which basically is, instead, like we have this whole mechanism, so we actually every small change uh, tr- creates a different color in some sort of way, But like what is currently done is that like, oh, all these changes, then you take the cells, you separate them, you go into like a whole different processes with equipment. You need to like kill the cells, wash them, disrupt them, and then you extract the RNA, which is a whole process using very expensive kits, and then you send it to sequence it. So you actually get like data, like very like you get a some files sent through the email where it's like, actually, oh, this is what the sequences say, like this is what it was happening. So for us, like the readout idea is actually to simplify the whole process. So you don't need all the other parts. You don't need to extract the cells, kill them, wash them, sequence them, like send them away to sequence them, which is also like a whole thing. Like I don't know, I come from Chile. Like for example, like sequencing. Like I mean, here in Germany, sequencing is really fast. But like for me, like we had to get the samples to sequence, and then we had to send them and wait for like 2 or 3 weeks to actually get results and to know what was happening. So I think like, yeah, I think like this idea of like being able to follow it on real time uh, is trying to make more to be focused towards more accessibility. I think like since our project is foundational advanced, it's more like something that we want to like help research be more accessible and yeah, and easier, I would say. But like I know, like if you go through iGEM, there's a bunch of different projects like that are trying to tackle climate change, therapeutics, diagnostics, and there is more like I don't know, like agriculture that are more like application based, like building a biosensor that we also find really interesting. But I think like in this year, like we ended up with this idea that I think we all really like a lot. But like I don't know, there's some other projects where it's like these biosensors where like you measure the levels of the contaminants in the soil and then they tell you that information. And so it's a lot happening. And I think like the idea of iGEM as a whole is also to try to make synthetic biology more accessible, more democratic, like to actually use this concept of engineering in biology, this synthetic biology, to push science forward and to actually have it to go beyond the lab and to actually have like an impact. And I think like what iGEM is doing is quite interesting, like in my perspective.
Nice, nice. Thank you very much for that. I think it makes sense what you said, because for me, one of the things that I did, like earlier in the episodes, was try to answer the question: how do we increase medical devices, or just medical technology, or just technology in general to like the third world, or to developing nations, etc. And one of the things was that the technology has to be so advanced that it's cheaper to distribute it, essentially. And that's what you've argued, that's what the point you've brought across, which I agree with. And in terms of the things that I wanted to get to, in terms of the ethical side of it, like Nika, to give you the chance as well, like what ethical considerations have you guys taken into consideration in terms of what are the potentials for misuse, what are the potentials for all these disruptive things that could happen, and what things you're doing in terms of making sure that the animals, humans that you guys use and test this on are well respected. So I don't know, Nico, like what would you want to tell us about the ethical considerations?
Mhm. So at the moment, our project is still not in the stage of the genetically modified cells actually leaving our lab. So all of that is like very well documented and regulated, and we have to destroy the genetically modified cells as soon as we don't need them anymore to do the experiments. But yeah, I think it's our responsibility as scientists and aspiring scientists to think about that type of thing. If I can give an interesting example of just what other people have done, for example, with synthetic genomes, where they would repurpose the genetic code to essentially only use a part of the information to code for the proteins, and not all of it, which would make it resistant to viruses. This was from the Church Lab. I won't go into details, but what they essentially did in addition to that, because those cells might have a competitive advantage over others because they are resistant to viruses. So if they accidentally leave the lab somehow, then they might just have a competitive advantage over all other E. coli that you could find in nature. So what they did, for example, was to make those cells dependent on an unnatural version of an amino acid. So they made it addicted to that specific amino acid, which is not present in nature. So once that bacteria leaves the lab, it does not have access to that particular amino acid and it cannot survive. So in synthetic biology, since we're already kind of engineering the whole system, we can add details like that to make sure that that it doesn't have unwanted effects in the nature.
Nice. So it's basically survival of the fittest, and that's what you alluded to. And basically, you're doing this using that mechanism of evolution to safeguard this technology. So, in case there's anyone there listening and thinking that, okay, if one of these cells leaks out into the world, it's gonna be like a zombie apocalypse. It's not gonna be happening. So, no, thank you very much for reassuring us on that front, Nika.
Yeah, I mean, this was not an example from our project, but this is just something that you can do since you're engineering the system, just to make a switch like that and to make sure that that everything is fine.
And I think it's important to add that RNA has not like a long life in the body. Like it's this messenger that gets destroyed, like this very like Mission Impossible kind of letter. So it's like it's sent, it does its job, and then it gets destroyed. So like and I think like that's also what we chose to focus on RNA, because like it's not you're modifying the DNA, you're modifying something that's gonna be destroyed in like a few hours. So it's basically this idea of like tracking, but like without the implications of like permanent DNA modification.
Nice.
That I think like it also speaks a lot to us, and our project has like a lot as I would say like every other project in iGEM. We are constantly talking to different experts from like I don't know like LMU to international people, and I mean it's not like we're like it's also like we're not in a garage or like in a lab like oh my god we're like doing this ridiculous. It's like we actually are. It's important for us to like talk to experts and yeah, you know, like I don't know, have this like we're not doing it like we're trying to be as responsible as possible. We're trying like to have every other consideration, and I think like for us also having people from different teams and talking to different experts, it brings this whole different perspectives. And that for us are really useful.
Sorry, Felipe, and I was basically saying as well, like, and this touches on a bit of like the myth, so the myth surrounding synthetic biology that exists right now, just like new things. The same way when AI came, people were a bit worried about it, and now it's basically a combination of AI and synthetic biology, which obviously makes some people sleep less at night already. So like the question is then, like in terms of like what's the biggest myth about synthetic biology that you think exists out there that people perhaps you could address, if I could ask it like that,
I
so
have an answer,
and
but
yeah,
it's
yeah,
probably going to be different than Nika's.
it's okay, both of
For
you can
me,
give the answers here.
yeah, for me, this idea that like, I mean, we have a lot of advances and like it's a lot easier than it is now. But like, I think there's this myth then like, in 5 minutes, anyone can like do like a bioweapon or like a radical like I don't know like gene editing, and I think like we're not there yet in terms because like even the people that are like doing like I think like at once like biology is not that simple like doing research like doing experiments is not that simple. There are a lot of like I don't know skills and knowledge, and like it's getting easier in terms of like it's getting more accessible to understand the concepts of biology and how DNA works and how RNA works. But like it's not that we're not at that point where like people can actually produce bioweapons in 5 minutes or or we're like, oh, now it's CRISPR, and now everything is solved, because like CRISPR still has a lot of problems that we're dealing with, and we need to tackle before like being implemented in a massive scale. So I think it's this risk of like simplifying everything to the genes and to the easiness, and I think like it's still very complex. It's still like it still has a lot of stuff around.
Nice, nice. And from your side, Nika?
Yeah, just building onto that, maybe also the connection of how fast the field of synthetic biology and genetic modification has advanced in relation to how fast the legislation changed. Because I feel there's a real discrepancy there, and that more scientists need to be more actively involved in the process, and maybe even connections between science and philosophy, or what Felipe is studying, which is essentially social studies and natural studies, and just to understand that gene editing is not what it was 10 or 20 years ago. Like what you used to do in agriculture was to do random mutagenesis, to just use chemicals to just try to completely randomly, without any targets, mutagenize your seeds, and then check which have a good phenotype, so which have the desired characteristics. And there you would have no idea what other off-target effects happened, and what was actually happening on a molecular genetic level. And now we have much better, much more targeted ways of doing that. But like Felipe said, it's still not perfect. CRISPR-Cas still has off-target effects, which we can then check by sequencing. And but still, just to I would like people to be more conscious of at least a few of the recent advances in the field, and how the technology actually works.
Yeah, and that is developed like there's scientists are working towards that, politicians are working towards that. Like it's I think like right now there's a real acknowledgement of the risk to like actually better regulate these technologies. I know, like when CRISPR was released, I actually don't know entrepreneur like the Nobel Prize winners. A call for a moratorium is like we have this technology that is wild. Like we call all scientists to not do uh, human permanent DNA modifications, and actually stop talk about it, decides what is good, what should be done with the technology. And I think as Nika said, like this um, dialogue between science, society, and politics is needed. So like actually legislation can actually uh, can actually be connected with what is happening in the field, so legislation doesn't appear when it's too late to do things already. And I think that's also to help it be more democratic, and to not I don't know, like a lot of research also is happening in what is called the quote unquote the global north. So it's like if you are allowing all these things to happening without legislation and without acknowledgement, like then who gets the benefits of all this? Like it tends to be like the same countries that always do. So I think like legislation and scientists and like a lot of people are do not want that, obviously. And I think like that's also like a good thing to look
Nice.
forward to.
Nice, nice. That's thank you very much for that. Really, I think it explains quite a lot. In terms of like the fears that exist, of either having this like super soldiers that are modified, or they they can run. I don't know how far and how long. And then looking also at the other side of the sort of like the legislation, because sometimes the science is faster than the lawmakers, is it? Because by the time they say, okay, we need to regulate this, it's probably 2 or 3 years after something is really kind of advanced and people are really kind of worried about that. So I'm happy that to hear that there is like a community that's sort of like working to make sure that we don't have that. In fact, the technology is not even really at that stage yet. So if people are worried about that kind of biological warfare, they shouldn't be worried, as the technology at this particular moment in time is not there, and
No.
there's already been regulations to make sure that we don't get to that point. And sort of like the last point that I really wanted to just know about from you guys is like, besides all the fears and stuff, like what do you think are the futures, or what do you think is the future of, like synthetic biology, CRISPR-Cas9 or CRISPR-Cas13? Like, what's the future? What do you think the future will look like? And I'm maybe to start. Yeah, we can start with you, Felipe, this time, and then we'll go to Nika.
I don't know. Like, I feel like it's such an evolving field, and I don't know. Like, 10 years ago, it wasn't what it is now, and I mean, I think it will go towards more applications probably, because like I feel like there's a lot of really cool ideas. If you look at the literature, there are really cool ideas that come from this like merging of engineering and biology, and I don't know. Like, I think like projects like what is done in iGEM have been showing different applications to like monitor pollutants. Like, I don't know, for me the use of synthetic biology, and I connect that a bit with my thesis. We use it actually for diagnostics, to do this like very low cost diagnosis checks that were like similar to PCR. That like when that happened during COVID, but like we have another technique. But like I think like using synthetic biology to make these diagnostics and access to these tools like cheaper and easier. I think like that's where I see a whole community working towards that to responsibly address the ways in which synthetic biology can address some things, and but it's also not a work only for scientists. Let's say like it's this like you need scientists and you need policymakers and you need philosophical and ethical dialogue, and I think like the field has been acknowledging that, and so yeah, I imagine that like synthetic biology has a whole potential in the future, but also like we need to keep having this conversation how to address it, and I think like they're happening. So for me, I feel hopeful.
Nice, nice. And yeah, Nika.
Yeah, I agree because synthetic biology has a lot of potential because, firstly, it combines a lot of fields. It combines, in a way, computational approaches with biology and systems biology, cell biology. So that's already really exciting. And I'm also quite excited about the future of AI, of the combination of AI and synthetic biology, because I would say already AlphaFold, for example, which a lot of people might know, is one of the, I would say, most successful and most exciting AI applications altogether. Because with AlphaFold, they were able to mostly solve the protein folding using AI. So essentially, before that, it was way more difficult to predict the 3D protein structure. And now we still don't understand how it happens, but we have a very reliable tool, a very reliable AI tool that shows us that. So I think there's a lot of advances in the combination of these two in the next decades, the
No,
next couple
and
of
just
years.
and just a quick question, really, before I let you go. How would you describe protein folding in a simplified way for people listening
Mhm.
who don't know what protein folding is?
Yeah. As we mentioned, the central dogma of molecular biology, we have the DNA, then we have the RNA, and then we have the protein. And the protein is essentially a chain of um, monomers called amino acids. And first off, it's just a linear chain technically, but then it folds into secondary structures called alpha helices and beta sheets. And then it folds into really complex 3D structures. And it's really difficult to predict because those interactions can be between really distant parts of the sequences. So just looking at the sequence, it used to be unclear how it actually looks in 3D. But with AlphaFold, you can really reliably predict how it works. And now with the new version of AlphaFold, we can even predict interactions between different molecules much better. So it's really exciting.
Nice. And just to add on to that, is that the reason why it's important for us to know how the proteins will be is because proteins make a huge part of our bodies. It's not just the protein, whether it's the creatine people get to get more muscles in the gym, etc. It goes on a much smaller level as when we're saying that okay, we need to because and again proteins are an expression of like RNA, and some proteins are generated because of the message the cells have received. So that's why all this is quite important. And I just wanted to add that basic knowledge there because it might be like, like university level biology, but for someone to understand even without this kind of a background. And now that makes a lot of sense. And just one thing that you mentioned before you go away as well that I wanted to ask, and just to also mention that people can also be reassured, is that the whole process of trying to add sort of like improve genetics, choose selective picking of favorite phenotypes, and by phenotypes I'm referring to either things that we see, and this has actually been happening for quite some time. And there was like it started with what they call like Mendelian genetics with this like monk like in the 1800s, and he did it with peas, for instance, and then they created this small diagram, like and then that's how people kind of started sort of like breeding certain crops. But this method took time because you'd have to do it over many generations to get like the one crop you want. It might take you like 20 generations to really weed out some genes you didn't want, and again, it's all randomized to a certain degree. And that's the power of synthetic biology, in the sense that we don't have to wait 20 generations to weed out something. We can try to do it like in a much faster way, in a much controlled way, once this technology reaches its full potential. So yeah, I'm really, and that's what I wanted to say, just to add on the basics as to how far these ideas have come. And I'm really happy to have had both of you today, like on the podcast. And I'm sure it's been quite informative for a lot of people. And I'll be putting the links below so people can also reach out to you. I'll be putting the dates for Paris. I'll be putting your LinkedIn links below. And so thank you very much.
Yeah, thank you for the invite.
Thank you
Thank
very
you for
much
the
for
invite. Yeah,
having
it's
us.
been really nice. Yeah.
Okay, perfect. Thank you. Take care.